Thanks for inviting me. And when I asked, what is the title of my presentation, I was told, you say what you want. So I had to come up with a title, and I figured, what is the message I want to convey? And I think it is, as we're looking into these pharmacogenomic medicines, that the learning curve that we face with every drug is probably, if anything, even longer than it is for conventional medicines. So let me give you the menu. Where can I operate? How can I operate this, please? Is there a mouse or anything? Ah, damn it. No, there isn't a remote. Anyway, let's... Thank you. Thank you.
Magic rodent. Magic rodent.
Magic rodents, yeah. So what I want to cover over the next couple of minutes with you is, first of all, pharmacogenomics is not black and white. It is shades of gray. When my secretary reviewed that slide deck, she said, Hans, are you nuts? You cannot show this title. Well, I'm still showing it, testimony to my innocence. Then we will talk about the learning curve, or the post-marketing evidence development. And I want to shed a short light also on the regulatory versus HTA post-marketing information needs, and then talk a bit about the toolkit that is required along the learning curve. So let's start with shades of gray. It isn't black and white. And it already starts with the level of individualization. We like to believe that pharmacogenomics is personalized medicine. Well, it isn't really. There are drugs that are truly personalized, like the autologous cellular immunotherapies. But I will not talk about these. I will confine myself to those where you have one drug and you're dividing it up among strata. And there are those that I would call multi-stratified therapies, and the best example is cystic fibrosis. I think you heard it yesterday. There are around 1,800 known mutations, so technically you could say there are 1,800 strata of patients. But the most frequent case that we have seen so far is binary stratification. And here the classic example is Herceptin, where you classify patients into one of two groups. So already here, we're facing shades of gray. But there is more to it. There was an interesting editorial in last week's New England Journal of Medicine about determinants of disease manifestations, even when we know a gene or genes that are operating in the disease. And it extends, of course, to treatment response. And the author said there are at least 3 factors acting on the disease expression and the therapeutic response. There is, of course, the environment. There is genetic background, usually modifier genes that we may know or may not know. And then what the author calls stochastic events. I'm not sure that is the right term, because what he calls stochastic events is just the unknown. And in 5 years' time, that stochastic event may be well understood if we can find out what are other modifier genes. But anyway, it shows that the biomarkers that we have today don't tell the whole story about a drug response. We pretend it does. We like to believe we can now separate the responders from the non-responders, but not the case. Look at the poster child, Herceptin. Depending on clinical setting, the response rate is somewhere, but it's always below 50%. Why not 100%? What happens to the other 50%? So some of these factors that are bulleted here must be at work. And here's another dimension of gray. It isn't necessarily so that when we have a biomarker, that it drives treatment decision. And I picked this because I found that in the literature, and I think it's an interesting case study. Some researchers found a single nucleotide polymorphism in the HMG gene. This is important in the effect of statins. And they said they could identify 2 strata of patients. One reaches a total cholesterol target level, let's call that a therapeutic success, in about 72%, and the other stratum in about 49%. Now, assume for a moment, and I'm not here to discuss the merit and the validity and reproducibility of that finding. Let's assume for a moment this is a perfect test, no misclassifications, it's wonderful. But is it a useful biomarker? Now, I would argue it is not from a regulatory perspective. We have a drug here that has a relatively benign safety profile if you dose it appropriately, and I would argue that the benefit risk is positive for both subpopulations. So it is, from a regulatory point of view, and I would say also from a physician point of view. If I was a prescriber, I would say this is a useless biomarker. It does not guide my decision. On the other hand, if I was a payer, I would look at this probably differently. I was in a meeting yesterday about value-based pricing and differential coverage. Now, if I was a payer, I would say, well, of course, the value that I get in a population in terms of bang for the buck will be much higher in the higher response stratum than it is in the lower response stratum. So it depends on who you are and how you look at information like this. So let's look at the learning curve, and let's start with the conventional drug development and licensing paradigm. The current paradigm under which we operate is learn, confirm, then license. And this really goes back to a clinical pharmacologist, the late Lou Scheiner, who said first you learn, you understand how your drug works, what dose is the optimal dose, which population, etc. You formulate your hypotheses, then you confirm those. That is what we usually call a phase 3 study. And then, if all goes well, you get a license. That is the idealistic scenario. Now, in real life, we know, of course, that very often we have to relearn and relicense, or in some cases, sadly, to delicense. But still, that is the ideal version. So if you follow that ideal paradigm, and you have before you a drug with companion diagnostics, so what we're looking here, a classic, what we call now pharmacogenomic or stratified medicine, then this is the ideal test to do. This is the granddaddy of all study designs in that space. It is the high road to evidence generation. And by the way, it is not a document from a regulator, it's a document from a payer. That's great. That's a 6-arm study with 2 levels of randomization. Trouble is, I'm not aware that this has ever been done, and we'll probably hardly ever see it in future, because it's hardly doable. The reality is a lot murkier. So when we come back to this idealistic scenario, and we look back at the experience, what we really saw was, in analogy to the 3 R's with animal experiments, these are my 3 R's for companion diagnostic guided treatments: retrospective analysis, retrofitting, rescue. That is the reality. And I don't blame anybody for that. We will continue to see those 3 R's, not because someone is doing anything wrong, but because our understanding of the biology of diseases isn't as good as we pretend it is. So we will necessarily learn as we go. So the learning curve will necessarily be a long one. And even if we like to believe that we have the right test, there will still be a lot of information needs to be covered in the post-marketing phase. And I have listed some information needs that go above and beyond what we would normally look at in the post-marketing phase. So you will not see here classic pharmacovigilance looking for rare adverse events. That's a given. I haven't even listed this here. But let's look at some of these aspects that we wish to learn in the later phase of the learning curve. Let's start with the test drug utilization. This is a paper I chanced upon, and again, here I'm not discussing the merits of this, and is this good or bad, or was it done well or not. I just invite you to look at those bullets here and read them quietly, and ask yourself, what is the question that these bullets here answer? Look at the first one. The testing, this is about Herceptin. The testing occurred in 88% of all patients for whom testing is recommended. So the question here is the test used? And it's up to you to say 88% is good enough or not. And then in the next bullet, we learn what was the ratio of negatives to positives. Why is that important? Well, you would here look at, is that positive-negative ratio similar to the one we saw in clinical trials? Here we see it's about 20 to 80. Let me exaggerate for effect. If in the clinical trial we didn't have 20-80, but 80-20, that should be a red flag, because very clearly we're now looking at a different population. And different populations may have different benefit risk. They have different environmental and other factors. So this is something to look at. And then lastly, these two bullets tell us, are the right patients treated? So this is something we want to know in the post-licensing phase, and it goes beyond what we have with conventional drugs. Here is another one. The test performance. So here we're going back to basics of the companion diagnostics. We know that they are lab dependent, and the positive and negative predictive value are population dependent. And so is the cutoff value in a given test, if it isn't just a binary expression. We will see, and we have seen that already, that while the evidence generated in the pre-marketing phase was based on one particular assay or test, that new tests or home brews will come up. There's nothing surprising here. And the problem is that these new tests will screen in or out, of course, different patients. And that will have an impact on the benefit risk and also on the cost effectiveness in real life. And then there is this term here, clinical utility. I've read a number of definitions, and we have a definition in our own guideline, and with all due respect to that, I would say this one, which comes from Janet Woodcock's article in 2010, to me is still the best. It's short and crisp and understandable. Is the test worth doing? Now, let me invite you to join a thought experiment. Assume I'm a drug company. I develop a drug and a companion diagnostic. Now, assume you're all a cohort of patients with a given disease. And I administer the test to all of you, and then I say those on the right-hand side of me now are test positive, those on the left-hand side are test negative. Now, I ignore those who are test negative, and I continue the development only in the test positives. And let's assume all goes well, and I have a drug that is a real life changer, very effective, perfectly safe. Have I demonstrated clinical utility of my test? What do you think? Well, no, of course not. I have not, because my drug might be as effective or even more effective in the test negative group. So I have not. But my other question is, assume that scenario. Should that drug get a license? Well, I guess everybody would agree if it's a life-changing drug, the answer must be yes. It would not be in the interest of public health to withhold it, even though we have no idea about the clinical utility of the test. So all of that will have to come in the post-licensing phase. So what we are seeing and what I have just described is really just this. In our own reflection paper, it's called Enriched Design. In the Australian document, it's called Level 3, Randomized Trial of the Drug Only. So that is probably the reality that we're going to see in future. And there is nothing wrong with that. Here is an example of how the post-licensing plan will depend on the licensing pathway right from the start. I believe you've heard this yesterday from Federico. I didn't have a chance to listen to all your talk, but question is, you know the pathway of this drug, ivacaftor. They have shown that it is effective in one mutation. They have shown that it is not effective in another one. And of course, we don't know about these 1,800 other mutations. You may be developing the evidence over time. And that is all fine. So even though you could say as a purist that the clinical utility has not been shown for all these substrata, so the answer is, well, yes, but you cannot ask for that in the pre-licensing phase. This has to come over time. It will be a drawn-out learning curve. And then there is this issue of relative effectiveness. This is coming to haunt us in the regulatory community more and more. Is that any different from a non-pharmacogenomic drug? Well, I would say some of the specifics may vary. For example, in the positive-negative ratio in a population may impact certainly the relative efficacy, also certainly the cost effectiveness, but I would argue that the fundamental information needs about relative effectiveness, or what our American friends called comparative effectiveness, are not much different when you look at one of those treatments or a conventional one. So let's now look at the information needs of the regulator as compared to those of a payer. And Marisa, in her introduction, said we have to look at the whole spectrum of decision makers. So what I've done here is I try to contrast some of those questions that come up in the post-licensing phase between regulators and HTAs. Are you interested in a companion diagnostic that guides a yes-no treatment decision? Classic example, Herceptin. The answer is yes, of course. Whether you are a regulator or a payer, the information need here is pretty much the same. When you have a biomarker for maximization of efficacy, it's already a different story. Please recall my example about the HMG mutation and statins. As I said, for a regulator, this may not be terribly important or useful, but if you are a payer, then it may be very useful indeed. The test drug utilization, how it pans out in real life, that is again important for both, as is the performance of the drug in combination with a new test, because of course the benefit risk may change, as does cost effectiveness. But when we come to relative effectiveness or efficacy, here again we see that regulators and HTA somewhat part company. We don't have it in our remit, of course, to look at relative efficacy. It's not in our statute, whereas HTAs and HTA bodies and payers are highly interested in that. So, lastly, what is the toolkit along that learning curve that we will need? And I don't want to complete this without applauding our own committees who are taking a very timely initiative by trying to draft some comments and guidance on the methodologies for post-authorization safety and efficacy studies, because in this space we will need that, probably even more than with conventional therapeutics. So we have a new document coming up, and there is also language in the reflection paper on pharmacogenomic biomarkers that we have on the web. So let me come back to this. This is the classic paradigm: learn, confirm, license. I posit that we may, in some cases, revise that and come to learn, license, and confirm some in the later phase, in the post-licensing phase. What that will require is a life cycle approach to research and licensing. It will require that regulators exercise flexibility, become more adaptive to new knowledge and information as it comes in, and it will also require that regulators and payers will collaborate to enable that learning curve over the entire life cycle of the drug. I will stop here and thank you for your attention.